To the organizers for letting me speak today. I'm speaking on force fields, Smirnoff, and the open force field group. Now, I seem to be unaffiliated in the documentation on the website, but I work for OpenEye. But the work I'm talking about today was actually done during my sabbatical, and I'll make a few nice comments about getting a sabbatical in a few minutes. But this work was done on my sabbatical with David Mobley's group, and this will come up later on. So this work that I'm talking about today is very exciting to me. I've been involved in force fields and coming up with charge models for many, many years. But this work that has come out of the sabbatical I did is some of the most exciting work that I've been involved in for a long time. And in my own mind, it certainly seems like a phoenix arising from the ashes. What I'll be talking about is a general force field for organic molecules and about this open and active force field effort. Now, what I'll be spending some time on talking about today is what the ashes, the whole idea is the phoenix arises from the ashes, and I'll be talking about the ashes, why we need a new approach, and then I'll be talking about Smirnoff, which is this new representation for a force field, and then I'll talk about the phoenix. The phoenix isn't the force field or the force field representation, it's the open force field effort, who and what it is and what lies ahead. So, the ashes. So the ashes of force fields. Well, we got to say, they're pretty good ashes, really. Force fields are super efficient. They can go faster than a speeding QM calculation. They can loop over large biosystems in a single second. Okay, and they are also somewhat accurate. They're parameterized to reproduce, conformational energies, and they can do that pretty well. They can fold proteins. You can do that with amber and charm and other force fields. And you can get a lot of small molecule geometries, correct, more or less, with MFF94 or any of the force fields that are around. And they've been the workhorse of computational chemistry in pharmaceutical industry for many years, but they do have issues. So this is the slide. This is where I should do the token trashing of small molecule force fields. I don't have time to do a really good proper trashing, so I'm just going to do like a symbolic token trashing. They're the whipping boy of current simulations now. It's what you can blame a lot of your inaccuracy on, and often that's true, but sometimes I wonder if it's always true. But I just want to point you to this really excellent article from Genentech by Alberto Gobbi, where he basically went into detail on where some of the defects are in force fields. But what I want to say today is that what we all know about the small molecule force fields is that although they do some things really, really well, they end up doing other things really, really badly. And this happens because you end up having these holes in your parameter, and you're always looking over your shoulder saying, are these happening for me with my ligand? Now, the old force fields at least would often let you know. They would say, you've got missing parameters, buddy. I am not proceeding on your calculation until you find me some parameters here. Whereas the newer force fields will often they'll just, proceed right ahead and give you some lousy calculations because the parameters are just bad. But what do we mean just bad? As somebody who's been working in force fields for a long time, just bad doesn't cut it. What is the issue here? And often an issue I'll be focusing on today is atom types, where the atom types are inappropriate. Or sometimes the parameters themselves are inappropriately generic. They were formalized to fit one kind of torsion, and that was good, but it doesn't fit every kind of torsion that atom type describes. Or it could be just a poor parameterization. It was done with at an inappropriate level of theory. But I think most of them fall into the first two. But I'm going to talk a lot today about atom types, and you can't blame all the inaccuracies of force fields on atom types, but I'm going to say atom types themselves, being atom types, is a lot of the problem. So I want to talk a bit about these atom types. So what are atom types? So atom types, firstly, you have some wizard who are the people who are constructing your force field, and they decide what the atom types are. And it should be pretty chemically evident. So Ca, which is an aromatic carbon, is around all these aromatic carbons. And you got a Ct for a tetrahedral carbon or an sp3 carbon around these carbons. And from the old days, this is how you did chemical perception. So atom types date from the inception of force fields back in the mid-20th century, and somehow they've endured to the modern day. This is how we do chemical perception, even in the modern day, with our force fields, and that's a little strange. It was state-of-the-art in the 1980s, but I think we need to do better. Another thing that's important is for most of the force fields we ordinarily deal with, because they've got all this great chemical perception in the atom type, you don't need to pay attention to things like bond order. So you know the bond between two aromatic carbons is going to be planar because they're aromatic carbons. Duh, it's got to be planar. And between the tetrahedral carbons, you're going to have that threefold torsion. So this has led to decades of frustration from me. So I just want to say, you know, I came out of the Coleman Group and I joined Merck and Company back in the early 90s, and I was pumped. I knew that this new force field that we had just come up with, that I'd been a part of coming up with, we could do small molecules. It said it right in the title of the paper. It did organic molecules. But then the first project I got on at Merck was cyclooxygenase 2. This is when cyclooxygenase 2 was a good target. And so I got to work on a lead compound there, which was flurbiprofen, which had this biphenyl linkage, which ends up being a real problem. And guess what? I could not work with this force field, this general organic force field. I had to come up with a few extra parameters. And then for the next compound, I had to come up with a few extra parameters and a few extra parameters. So 18 years later, we had this force field, this small molecule force field. This predated GAF, so I had to come up with it before GAF. And it consists of almost 3,000 parameters. And this is how we actually ended up being able to do our calculations on our small molecules. So here's what was the problem with the biphenyl. So here's flurbiprofen in here, and here are my atom types with the carbon alphas. The problem is this bond right here. It's two carbon alphas joined together, but it's a single bond. It is not a double bond. It's not even really conjugated. So with the atom types, their implied bond order, it says that these two rings have got to be really planar. Well, we know that a biphenyl with that almost single bond has the two tilted at about 40 degrees. So much for the atom types, but that was okay. I was one of these guys out of the Coleman Group. All I had to do was make a new atom type, which I did by making a CP atom type. Oops. And this would actually help me here, because I could just make that a single bond, and this solved the problem. And the issue is now just that, you know, this new atom type, supposedly to get my chemistry correct, is just to fix up this bond order defect. So I say this is a miscarriage of atom typing. In the same project, we then had this new series which became atorocoxib, which is actually a COX-2 selective inhibitor from Merck sold in Europe. But so you had the two CPs across that bond. But when you put the two CPs across this bond, now your aromatic bond with the CP-CP single bond was through something which needed to be a double bond. So once again, we drove into the pit. So the solution was more and more atom types. So then that led to what we have, the current situation in many, many force fields is you've got this multiplicity of parameters. And I just want to show you all these parameters are actually, they're a bond angle parameter, and they're all the same parameter. In fact, there are hundreds of these parameters in, redundant force fields, and oh boy. And in permafrost, there's 920 of those identical valence parameters just to cover all the different atom types we've made, and there's 60 for CP alone. And this is a valence angle, which wasn't even why we made it. Then over here we've got in GAF, there's many, many identical torsion parameters. It's the same problem. So this, and then when you do that, you still end up with some problems after that. So here is, you've got CPs and CQs, you've got how many different torsion parameters, who knows. But when you do the dynamics, and this is with the modern day GAF force field, still dynamically you have this unnatural behavior because not all the different combinations of atom types in the torsions have been covered. And so the central ring buckles. So this leads us to, so I just want to summarize my trashing of atom types here. This leads to the force field frustrations where we have this chemical perception through atom types, this old world way of doing things, leaves us relying on expert intuition and hand tweaking. You've got real big difficulties to come up with a force field that actually works. I think it was a poor choice for a data structure in the first place, it was all they had. You had this excuse back in the mid-20th century. We don't have that excuse now. And you have got these massive redundancies in parameters like I was showing you. And these masquerade as independent parameters. So when you've got these automated parameterization schemes, they're fitting these slightly different parameters to these supposedly independent formulations, when actually it's simply a redundancy and they should be constrained to be the same. So this means that making force fields is a monumental effort, taking many person years. So did I complain about this? Bitterly. And some of the people I was complaining to was the three amigos, David Mobley, John Chodera, and Mike Schertz. I'd complain at GRCs, I'd complain at the ACS, I'd complain at the sample meetings, and we all commiserated and couldn't do anything about it. So then the company I used to work for closed, and I joined OpenEye, and at OpenEye, two good things happened. I'd say more than two good things have happened at OpenEye, but I want to talk about two today. One of them is that I was forced to learn smarts and smirks and smiles. So at Merck, they'd had a different chemical perception scheme, but I learned this scheme. I learned this really great scheme of embodying chemical substructures in a line of text. So this, you can apply this directly to a structure, and it will identify substructures that include bond order and various properties of the atoms. So I realized that this is direct chemical perception, whereas these atom types have been kind of a secondary one where you do atom by atom and you paint an atom type on and then you connect them together. Why not do it directly with a smarts string? The second good thing that happened at OpenEye was they have sabbaticals, which was great. And my one came up a couple of years ago, and I went and did my sabbatical with David Mobley's group on a project that included John Chodera and Michael Schertz in their groups, and then together with them and with OpenMM, we actually came up with this scheme of using smarts. Smirks is the only part of smirks we include is the annotation to identify particular atoms that we're applying our parameter to. So we were able to come up with a force field incarnation which used this scheme. And what happens, so here's the scheme for the torsion in a smirk string or a smart string for this very tetraphenyl that's causing the problem, and the only difference between the normal aromatic double bonded parts, or 1.5 bonds, and the single bond is the embodiment of the bond order. And with these two different very traditional looking torsion terms, it just gets the tetraphenyl correct because it captures the bond order. It gets it right without even trying in 2 lines. No special treatment. So we generalized this to come up with a new data representation for a force field called the Smirks Native Open Force Field, which abbreviates to Smirnoff. And this new force field representation can be used directly to represent your force field. So this is the Smirnoff force field for methanol. And basically, it's just copying over the parameters from amber. But because we've been able to take these what used to be done with atom types and just formulate it in smirks, and it's done in a very few lines. And I want to point out two particular lines. It's maybe too tiny to see, but there's a line up here with a particularly long definition of an oxygen, which we needed to get the bond length specified. And then down here in the non-bonded forces, there's a definition of an atom, a hydrogen atom, which says it's bonded to a carbon atom, and that carbon atom is bonded to one of these other lists of electronegative atoms. Now this is an embodiment of what amber has as the H1 atom type. But essentially, we can have it down here just when we need it for the non-bonded force without having to copy it everywhere else. And this means that we don't have to have all these redundant parameters. So as my last act of my sabbatical, I went by and I took all the force field and the small molecule force field we'd had with permafrost from the Merck small molecule force field, which is built on top of Parm 99, and I took those total of 3,600 parameters and was able to embody it in a Smirnoff built on the Parm 99 and the Frost force field. And I just want you to notice that I was able to, it could just condense down into less than 1/10 the number of parameters. And this is almost entirely due to the removal of redundant parameters, although I did telescope together very similar parameters that only had these very slight differentiations, which I think were just due to the having artificially independent parameters, which weren't really. So we've got a small molecule force field that consists of 332 parameters. Well, what can you do with 332 parameters? Like, what do I mean a small molecule force field? Well, essentially, the chemical coverage we can get from that as we try and evaluate drug bank and zinc and E molecules, we can get greater than 99.5% of those molecules parameterized. Now, I'm not sure how great the parameters are, but the point is within these 332 parameters, because we use this more compact data representation and we just skip atom types completely, we can get the whole thing covered with a very few parameters. Interestingly, the parameters that were in the original permafrost end up only being able to do less than 2/3 of the two smaller databases, because using atom types, they came up coming up with missing parameters because there was some combination which I was missing. This is the plague that we've suffered with for many years. So this is a force field which can be used to calculate small molecules. Well, so who can use this? Oh, actually, before who can use this, so how good is it? We looked at FreeSolve, so David Mobley's lab, who came up with FreeSolve, the compilation of most of the experimentally known free energies of hydration. This is with Smirnoff, and this is how well we produce the free energies of hydration, and this is GAF. So we do pretty much as well as GAF. Here's the Smirnoff versus GAF. It performs pretty well as GAF. And here's a bunch of neat organic liquid densities. And again, we have GAF on the left-hand side and Smirnoff on the right-hand side. We've got one outlier here where Smirnoff does particularly badly compared to GAF. Fortunately, it's water. And we wouldn't want to use either Smirnoff or GAF models for water. We've got much better models for water. But except for that, the Smirnoff 99 Frost is performing almost as well as GAF straight out of the box with a fraction of the number of parameters. So how do we use this and who can use this? Well, you have the Smirnoff 99 Frost, you have this XML file that expresses the force field going into OpenMM on the one hand, your system going into the OpenMM on the other hand, and you produce an OpenMM object. Well, not all of us want to use OpenMM to do our simulation, but fortunately with Python modules and GitHub, through either Paramed or Intermol, both available as Python modules through GitHub, you can take this OpenMM system and translate it into input files for any of these molecular dynamics engines. So this interoperability, I just want to mention, I'm not talking about any other OpenEye stuff, but I'm just saying this mentality is underpinning our implementation of Smirnoff specifically and MD in general into our cloud modeling environment called Orion, where we're basically building in this interoperability so that a user is not wed to one particular molecular dynamics engine, but actually can take this technology through to many through this, this portal of Paramed or Intermol. So I just want to conclude this section on Smirnoff by saying we've got this paper coming soon, but we've got the manifesto out as a preprint, which is just basically a much longer version, everything included version of the paper at this DOI. And it basically is summarizing what I've said in my talk to date. We've got Smirnoff as this new representation of a force field, which is free of atom types. It's just you don't have any atom types at all. And you've got this Smirnoff 99 Frost, which is a working example, which is useful now and actually gives pretty decent results. And the paper goes into some future directions, which I won't go into today. So now I want to get to the open force field group, the phoenix arising from the ashes here. Now, what is the open force field group? It's more like who is the open force field group? And the open force field group consists mainly of these groups here, the groups of John Chodera, Mike Gilson, David Mobley, Michael Schertz, and Liping Wang. So these are the academic groups that are doing the heavy lifting. They're keeping me around. They've got Kenneth Cronline from NIST, and they're keeping me around, I think, mostly because I've made every mistake that's been possible to make in the development of force fields, and I want to try and help them to not do the same thing. So within the context of this effort, these are the goals. So I just kind of cut them and pasted them as text in there because I can't spend a lot of time on them and I don't really know how to paraphrase them. The first two bullet points have to do with providing force fields to people. Notice that there'll be open and versioned force fields, and we're aiming for consistent improvements. We're not going to produce one and say, this is the mother of all force fields. We're saying, this is where we've got to now. We were improving these things, and you'll know what's improved because from the second bullet point, you'll have built-in estimates of confidence in the predictions. The last three bullet points have to do with building force fields. We also want to share with the community the tools that we're making to help parameterize them and the data sets that we're developing to parameterize them and to help the whole community move forward on this front. So what is open? You've got a guy working for a software company up here talking about an open effort. So what does he mean, open? So this is what we mean. It's open. All the tools are open source. It's on a GitHub site, which you can visit now. The data is open. We don't have as much as that now, but we're collecting that and curating that, and that will be made available. And the science is open as well. As we make our advances, they end up being, going into GitHub and being available to the community, and the force fields will all be open. It's about as open as I can think of. And this is what it looks like on the site. So right now we've got 6 projects posted, and I just want to spend the remainder of my talk showing you what happens with these projects and how you can use these projects and what they can do for you, and maybe a little of what they're doing for us. So I want to focus now on these two. So Smirnoff 99 Frost, that's the project that holds the force field, and this has some tools for using the force field. So how do we use that? Well, here are the 6 lines of Python that you need to get your molecule parameterized by Smirnoff 99 Frost and into your simulation. So from the first 2 here, we're importing some tools from the open force field project. And then you get your molecule in there, you get the topology, and then you apply the force field here. So this is applying the Smirnoff 99 force field to your molecule. And then you produce this thing called system, which is produced as an OpenMM object. So that's an OpenMM object. Well, great. What are you going to do with that? Well, that can then just go straight into Paramed or Intermul to become the system if you want to use one of these other engines. We're going ahead and using it with OpenMM because that's what we'd like to do, and we actually like OpenMM a lot. So I just want to also talk about, now these are about the parameterization tools that we're building. So here I'm not going to spend very much time on this slide. I just want to show that we're going to be using Bayesian Inference, a lot of science championed by the Chodera lab, where we can use Bayesian Inference to try out different parameter sets in a simulation to calculate an observable, and the observable could be things like the hydration free energies or the densities or static dielectric constants that we're getting very good measurements from, experimental measurements from through NIST, or it could be torsional potentials that we're getting from Psi4. Another thing we're not turning up our nose at all, but willing to use is the least squares type fitting engine from Liping Wang's lab. So both of these we'll be using for parameterization. So where does Smirnoff 99 Frost come into this? It is only a starting point. It's a good start. It's our zeroth order force field from which we will perturb to make better force fields. And there's a bunch of things we've got on our list of work we want to do to improve it, going from easy down to hard. So we're also going to do this, basically more or less chronologically. This reflects when we're going to try and address these things. The point about the blue box and the pink box is that with the blue box, we think we can maintain compatibility with the amber force fields. That's kind of the parent force field for Smirnoff 99 Frost. But then at some point, we're going to break amber compatibility to begin to look more deeply at some of the functional forms and go in a new direction once the effort gets going. But at all points, consistently, as we go through, we'll be coming up with consistently improving versions of the force field that will be accessible to all. So I'm going to spend not very much time on this project here, Smarty. This is my favorite part of this whole open force field effort, although I shouldn't play favorites. It is my favorite part, and it is really, really exciting because I think it's going in a new direction, a direction where the science has not been able to go before. A few moments ago, I talked about Bayesian inference of parameters. Normally, think of the parameters, the numbers that drop into the functional forms that express the physics of your force field. But I want to mess with your heads a little. What if the parameters that we're changing is actually the chemical perception? What if you take this string, which is a smart string, and you consider that all the different parts of this smart string, like X4 means the connectivity, there are 4 connections to the carbon atom, What if you take the, you've got these hydrogen atom counts and these other properties that are part of a smart string, and what if in fact you sample different chemistry by changing these different properties in the smart string on the different atoms? That way you actually change the chemical perception of the parameter. You are sampling the chemistry. That is what is going to go into some of this force field fitting as well, to see how we reproduce the observables. That is really exciting. That is something I've never seen done before, sampling the chemical perception. And in fact, in some very exciting work, which Caitlin Bannon and David Mobley's group has been a central pillar of, it's carrying forward learned chemical perception of the force field typing rules. So this is another preprint which is very freshly out. It actually delivers on the proof of concept of this, and it shows that with smarts and smirts-based chemical perception, you can get the chemical perception of that for a Smirnoff force field to be learned via Bayesian inference. So this was on basically pretty toy problems, but we were just trying to see if we could do it, and we can. And that is really amazing. That, I think, is how we're going to carry forward a lot of the effort. It's very exciting to me. Imagine being able to do the machine learning of the chemical perception, and then at the end of the day, you'll have smile strings which you can actually see and interpret and understand. Very, very exciting. So in closing then, I just want to mention a few words about the consortium. So this here is a screen which is hiding a lot of logos of some industries which are interested in collaborating with us. So Bayer and GSK have both agreed to peek above the screen so we can talk about them. They're interested in helping the open force field group go forward, and through an intermediary of the Molecular Software Sciences Institute, they will be helping to support the work of the 5 principal investigators of this effort. So this industry effort is going to help fund the open force field group, and it's still coming together. It's still open. Ross Walker, where are you? Where is it? Here's Ross. So Ross Walker, he's been our liaison through GSK, and you're available if people are interested in being part of it.
Yeah, also Katerina. And Katerina is...
Katerina, oh,
yes.
Katerina as well, that's right. So I just want to, and so this collaboration is still open if people are willing to join. So my conclusion slide is with this open force field consortium, we're coming up with a general force field for ligands, biopolymers. And as a new approach, I'd say its pillar is we start with being able to escape atom types completely with this Smirnoff representation. We're getting a broad, diverse data set for parameterization. I haven't said anything about this really, but this has a lot to do with our collaboration with NIST. We're going to have automated iterative parameterization with these tools that are using Bayesian inference, as well as just the least squares fitting kind of approach with force balance. And very excitingly, we're going to be treating chemical perception as a parameter. And it is open. Oh yes, the lunch announcement. So we're going to have an open force field lunch get-together tomorrow. So if people are interested, are involved in the collaboration or are interested in the collaboration, tomorrow afternoon, please grab lunch. Then we're going to form an aggregate around David Mobley in the lunch area, and David will take it from there. And with that, I want to conclude just by thanking the many people who've been involved in this effort up to date. There's new people coming on, so I might even be missing a few names on this slide. And it's really, I'd say, the most exciting work I've been involved in a great many years. Okay, thank you.
Questions for Chris?
Yes, Chris, maybe I misunderstood, but where you showed the comparison between Smirnoff and GAP, if I understood it, you just took GAP parameters and basically reduced them down with a Smirnoff's approach, right? So surely the results should be identical.
Well, it was actually permafrost. So back in my decades of frustration, I was well along developing this other force field before GAF even came about. And the early GAF made biphenyls planar. Right. So I was way past that by that point. So there was a divergence that happened quite early. So Smirnoff 99 frost does not come from GAF. There's been I'd say Caitlin has added a few parameters later on to fill in some particular holes, and we took a few parameters for GAF, but really just a small
So
handful.
if you took GAF and put it through the Smirnoff approach, you'll get identical results, where you can go back and forth without history.
Well, actually, where we would not get identical results is where you get the bugs. So actually, we uncovered a couple of bugs and torsions from Parm 99 Frost. We couldn't reproduce Parm 99 Frost with the Smirnoff representation until we actually had these very complicated smart strings to make exceptions, turning a particular torsion back into the unwarranted generic for the torsion, because back in Parm 99 it hadn't been actually selected out. So remember that central ring was buckling in the tetrophenyl dynamics of GAF? It would be actually very difficult to get Smirnoff to do that, because you'd have to create exceptions, turning aromatic bonds, which should be planar, non-planar.
I think partly what you're getting at is whether hydration-free energies are really an interesting test. And so partly that just verifies that we didn't screen anything out too badly in the compression and conversion process.
Right.
So yes, thankfully we didn't screen anything up too badly.
Right.
Any other questions? Yeah.
So have you thought about addressing the compatibility issue? So you're developing a ligand force field. You're beginning to develop the force field itself, right? But how do you make it compatible with, say, charm or amber?
Well, the low-hanging fruit here is amber. It's going to come out of the box actually pretty compatible with amber. Again, within the OpenMN formulation, you can actually take in an amber force field or a charm force field and apply it to the protein part of the molecule, and then use Smirnoff for the ligand part of the molecule, and then the non-bonds just have to communicate. And in terms of the compatibility there, at this point anyway, it's very compatible because they're pretty similar non-bonded terms. The charges, of course, are done separately, so it's really just about the non-bonds at this stage. If you had a covalent ligand, there'd be an issue, but as somebody who's been involved in the field for quite a while, right now, if I had a covalent ligand, I'd put all my money on Smirnoff.
One more question.
So now that you have a sort of 300-line parameter list, do you have a feeling for how many, for example, torsional scans you have to do to completely reparameterize that?
Well, the interesting thing about the torsional scans, because this is, of course, a key question for us, and we are in progress right now, beginning to look together at the torsion scans. And one of the marvelous things I can confess to, as somebody who's been involved in parameterizing for a long time, is I don't know how many extra torsion terms we're going to need. These generic terms are going to have to break apart into child terms when they're needed. Back in the day, I had to put on my wizard hat and guess when they were needed. Now we're going to have Bayesian Inference working on a great many molecules which will tell us when they're needed, or test when they're needed, and if they're needed, they'll keep them, and if they're not needed, the Bayesian Inference will throw them away again. And I don't know how it's going to turn out. It's going to be very exciting.
Okay, let's thank Chris again.